Hi class, and welcome to our second portion of Organic Molecules Part 2. We're going to be talking about proteins and nucleic acids, the second part. Remember, we had 4 organic molecules that were really important. We already did lipids and carbohydrates, so now we're going to start with proteins, and then we're going to follow up with nucleic acids at the end of this video. So let's start with the protein. The protein has a polymer, which is called a polypeptide, and the amino acid is the monomer of a polypeptide. Now let's go back to that polypeptide word. Polypeptide is not just the name of the polymer, but the polypeptide can mean just one protein. But polypeptide generally means just the string of amino acids. And then when we put many polypeptides together, we actually get the protein. So think of it as the polypeptide being the yarn you need, and then putting many of those together, and then you eventually get the scarf that you need to be worn, because every protein is going to have a function. So if we look below here, we see this structure, and what that structure is actually an amino acid, and we have this carboxylic acid group. We actually do call that a carboxyl group, not a carboxylic acid group, and we have the amino group. So the amino group is where we get the namesake from. We have the carboxylic acid group, and then we have right here, we see the R group. Now the R group is not actually, quote unquote, any type of element that is an R. Actually what it is the thing on the amino acid that changes, because we have 20 different amino acids. So this is everything except the R stays the same from amino acid to amino acid. So we have 3 different functions for our proteins, and the first function is structure. In our muscles, we need structure, and we get that from our proteins. We use the protein myosin to contract our muscles, and we use it for defense in our immune system, and we use antibodies like IgG, IgA, all of those. We use those for defense against bacteria, viruses, anything that invades our bodies. And then we also use them for receptors and transport, cell-to-cell communication, transporting oxygen, transporting nutrients, anything we might need. So that is what we actually use the hemoglobin molecule for. So as you can see, we have the hemoglobin molecule right here, and we actually do have multiple polypeptides in this that we are using in this hemoglobin molecule. So there are all different types of examples for each of these different functions. Now, how are these proteins made? The same as your lipids and your carbohydrates. Every one of these is made with the dehydration reaction. But this time, we're going to link the carboxyl group of the amino acid to the amino group of the next amino acid. So we're just linking them in the same fashion, in the same order. That way we don't get all mixed up. And the covalent linkage, or the covalent bond, is going to result in a peptide bond. So that's a new term, a new name. So make sure you get that down, highlight it. And this new term is the peptide bond, and the peptide bond only occurs in proteins. Now, this is just a look at that dehydration reaction in these amino acids. If we can look at this, we have the carboxyl group here and the amino group here, and we're actually taking that hydroxyl from the carboxyl group and the hydrogen from the amino group. We're grouping them together, and we're taking the H2O, or the water, out, and we're creating that peptide bond. Very important. So structure fits function. In proteins, amongst all of the macromolecules, this is the most important, because a protein is very specific to its function, even more than any of the other macromolecules. All macromolecules are their structure is specific to their function, but with proteins, its structure is the most specific to its function. So a protein may contain one or many polypeptides. We've already talked about that. And the structure is very specific to the needs of the function. So if a structure gets disrupted, so does its function. So this would just be like you use your hands to tie your shoe, and if some accident were to happen where you lost a hand, you would have to figure out how to use just one hand to tie your shoe. So if the structure gets disrupted, so does the function. So the wrong shape equals the loss of function with proteins, and that's called denaturation. It's caused by a change in the protein's environment, which may be the pH, it may be the heat, it may be the salinity. And it doesn't necessarily have to be too much, it can actually be too little. And you actually do this every single day. You denature things every single day. You do this in your kitchen when you cook. You might cook eggs, you might cook meat, anything with protein in it that you cook, you generally are denaturing it to be able to eat it and ingest it. So just think about that the next time you're cooking. You are denaturing those proteins so you can cook it and safely eat it. Alright, so we have 4 levels of proteins, and the first level is called primary structure. It's just that amino acid sequence. Remember I said we had 20 amino acids? It's that sequence that we have. Now, if that sequence gets disrupted, even just by one amino acid, it can actually cause a lot of harm. And that harm, for example, could be in the form of a disease. Sickle cell disease is caused by the change in just one amino acid. So it's really important that our bodies get this amino acid sequence exactly correct. Once we have gotten our amino acid sequence down, our bodies have made that amino acid sequence, we then go to the secondary structure. The secondary structure is coiling or folding of the polypeptide chain. So we started out with that primary structure. We made the polypeptide chain. We made those peptide bonds in between the amino acids. And now we're actually getting a little bit of shape. We call the coiling the alpha helix. And as you can see, these red dots here are actually hydrogen bonds in between the amino acids, which is going to help it hold the shape. And in the beta-pleated sheet, we have those hydrogen bonds again. So if it's folding, it becomes the beta-pleated sheet. Now next, we have the tertiary structure, which results from interactions between the R group of various amino acids. So if it's interacting, it has different things that could happen. One of the examples of something that might happen is a disulfide bond or bridge. If you have curly hair, you have disulfide bridges in your hair. Now this is only for naturally curly hair. If you use a curling iron, you are not creating disulfide bridges. It's just if you have naturally curly hair, these do occur in your hair. And then the last level is quaternary structure. This is where we have 2 or more polypeptides that associate with each other. As you can see here, we have 4 polypeptides. That would be the case with hemoglobin. Now, another protein that we see a lot in our bodies is a protein called collagen, and that resides in our ligaments and muscles, and that's what gives us the ability to have elasticity in our muscles, and it allows us to have stretch. It also gives us a little bit of structure and strength as well. And that actually has 3 polypeptides. So different proteins are going to have different amounts of polypeptides that associate with each other. So just to review, the primary structure is just the amino acids. The secondary structure is going to be that alpha helix or that beta-pleated sheet. That tertiary structure is just going to be the interactions in between that polypeptide. And then finally, you're going to have more than one polypeptide with the quaternary structure, which finally is actually going to make your protein. Because remember I said you don't necessarily just have one polypeptide. You have more than one polypeptide to make a true protein. So now let's talk about nucleic acids. Nucleic acids are exactly what you know them to be, their DNA and RNA. But the polymer is called a polynucleotide, and that would be because a monomer is a nucleotide. So remember that word, that prefix poly means many, so we're going to put many nucleotides together. And DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. Now, the sugar in DNA is deoxyribose, where in RNA it is ribose. So you can kind of see that in when we write out the full word. But make sure you take note of that because that's going to be important when we get to the monomer here in just a second. So looking at that nucleotide that I just talked about, we have a phosphate here, and that phosphate does not change between the DNA and the RNA. That stays exactly the same. But we do have right here a sugar, and that's a simple sugar. That's a monosaccharide. Remember from the last video we talked about monosaccharides. But that does change. Remember I just said in DNA we have deoxyribose, in RNA we have ribose. So there's a slight change in these, but it's not a big difference. They do stay a monosaccharide. And then we have our nitrogenous base, which is going to be what actually kind of stores our information. So in nucleic acids in DNA, we have the bases A, T, C, and G. It's not important for you to know the names, so don't really worry about that. And in RNA, you see that we do not have T's, but we have U's. The rest of them stay the same. And a particular nucleotide sequence, so those A's, T's, C's, and G's that can instruct the formation of a polypeptide is called a gene. So this is the biggest part of what makes us us. This is what instructs what is going on with us. This is how we know who we are because of this DNA. So let's go ahead and move forward and talk a little bit more about these nucleic acids. Okay, so DNA is double-stranded, and everyone, this is a little bit of a review for you, that it is double-stranded, but you can see that with these double strands that you have this circular thing, and then you have this blockish thing. So what we are seeing is actually part of the nucleotide. It's the sugar and the phosphate repeating on each other. And you can kind of see the A's, C's, T's, and G's here jutting out from it. And those are just your bases that are holding the information in. It is a double helix in shape. And in the DNA, we have one hydrogen bond in between A's and T's. A-T spells at, and it's a two-letter word. Or we have two hydrogen bonds, I'm sorry. You have A-T, it's a two-letter word, so it has two hydrogen bonds, and C and G has three. So we do have hydrogen bonds there. And then in RNA, we have a single helix. We still have our nitrogenous bases. They do not bond to anything, but they do help make our proteins, so they are very important. Okay, this is the end of our macromolecules. We will talk a little bit about enzymes and their role in macromolecules, but make sure you've got good notes, lots of figures, and we will talk about this later in class.